Thank you very much, Marileen, for agreeing to give a build a cell seminar, and you can get started.
Okay, let me share my slides. Yeah, this looks okay again.
Perfect.
So you should just see many pictures of the same people over and over again. And let me make this a bit smaller. I will not see you right now, but I would suggest, since for me it would be nice to keep this a little bit informal. I know there's 40 something people online, so I don't know how that works lately. But please feel free to stop me and ask questions. If not, I just show what I wanted to show. And then we have questions in the end. The only thing is I cannot see if anybody is, asking a question, but maybe you can, Kate.
So if someone puts a question in chat, would you like me to speak up and read you the question?
Yeah, or just ask people to unmute and then ask me the question, whatever. I haven't been to the, whatever works. If it's
Will do.
Yeah, just let me know, because the only person I see is you.
All right.
Yeah, so what I thought I would do is have really a work in progress talk. So what we are trying to do, and give you some examples of what didn't work so far and where we are hoping that it might work, which is all about building a minimal DNA segregation module, I've called it here, for synthetic cells in the context of the larger consortium program that we have running in the Netherlands. So I'll start, I've given a talk here before with a little bit of context where we are coming from as a group. So the group you see here is at various points in time in Delft, in my garden, and actually on the lower right is just last week when we had a group trip to France. So we're sitting on a boat in Lyon here somewhere. And yeah, so just let me start by explaining that we have a long-standing interest in the cytoskeleton, and also in reconstituting cytoskeletal processes from the bottom up ever since, and I'll show a few slides about this. I'm running my own group, and this is sort of an example of how far we've come already several years ago in building eukaryotic mitotic spindles in confined spaces, in this case in microfluidic droplets, even though in the last years we've also spent a lot of effort, I'm not showing that, in making minimal eukaryotic cytoskeletal systems like spindles in vesicles. We actually have quite a bit of problems with that, because encapsulating tubulin in complex buffers with other cytoskeletal proteins is giving us a lot of trouble, but I would be happy to talk about that in at some other time. But this is just to say that starting from the bottom up, trying to understand how cytoskeletal systems get organized in confined spaces in the cell, and how this contributes, for example, in complex spindles to the segregation of chromosomes. That has been for a long time our interest. And I'll show a bit more about this. So this is over the years we've come to build what I called functional cytoskeletal complexity. So very early on, and we still do that, we're interested in studying the dynamics of individual microtubules, how they generate forces, and we've used in the past, and are still doing that, microfabrication techniques to create artificial barriers that mimic something that these dynamic filaments may run into, like a chromosome or a cell membrane, and then study how this affects the dynamics and how forces get transmitted to different objects in the cell. And I'm going over this very quickly, so if somebody wants to ask anything or know more, please stop me. And in addition to understanding the dynamic and force generating properties of individual filaments, we've also, since many years, encapsulated these systems in confined spaces like droplets and vesicles, where we're interested in how does a single microtubule ester position itself, depending on the parameters of the dynamic filaments, and how can this be also controlled by associated molecules that interact, for example, with the ends of microtubules, but also that interact with the cell boundaries, like molecular motors that get localized to the cell periphery. So it's a combination of mechanical aspects, biophysical properties, biochemical control of these properties, and how that contributes to self-organization in cells, for example, in the context of a spindle. I'll show one more slide in a second about what different people in my group are still doing in that direction. But then, since we've been interested in also contributing to this consortium of building a synthetic cell, we've switched some of our effort towards designing and building a minimal active DNA segregation system for synthetic cells, which, as I will show, is much more inspired by how bacterial systems, unicellular systems, segregate DNA in an active fashion. So that's what we are now, part of the group is working on. And that's where I'll spend most of the time showing what we're trying to do and how far we got. Like I promised, just introduce the people in my group that are working on the eukaryotic side of the cytoskeleton. So we still have some efforts trying to understand how post-translational modifications affect mechanical properties of dynamic microtubules. So this is Cinda, and she's actually a PhD student shared with the lab of Karsten Janke in Paris, who is a specialist in what's called the tubulin code. And the tubulin code is, refers to post-translational modifications, of which there are many in tubulin that affect not only the mechanical properties and dynamic properties, we think, but also how different associated microtubule proteins interact with microtubules. We also still have an effort in understanding how forces get transmitted between shrinking microtubules in this case and proteins that are found at the kinetochore of chromosomes. So chromosomes have a specialized protein complex in eukaryotic cells called the kinetochore. And there's many proteins involved in that, but the protein sort of closest to the microtubule interface, one of them is called NDC80. And it's a protein that interacts with shrinking microtubules such that microtubules can generate pulling forces on chromosomes. And one of the questions we're asking is how many of these NDC80 molecules do you need to reliably transmit force between the shrinking filament and the rest of the kinetochore? And we use DNA origami nanosprings as a force sensor. So we can stick these nanosprings to the surface, then hook them up with a controlled number of NDC80 molecules, again using DNA origami, and then use fluorescence microscopy to see how these nanosprings get stretched to about a micron maximum, and use this as a way, a bit more high throughput way to measure forces compared to optical tweezers, for example. And then we also still have efforts, like I said earlier, trying to to move these minimal spindles from microfluidic emulsion droplets to vesicles, which is what Yash is now defending his thesis on at the end of the year. And like I said, it's not an easy task. And then the last one is we have still collaboration with several specialists, or actually it's relatively new specialists with cryo-EM, but also liquid phase EM, to get a more molecular view on how molecular interacting molecules that you find at the end of microtubules, how they cell organize, and how not only how are these molecules organized in supramolecular complexes at the ends of microtubules, but how is this then related to control of their dynamic properties? So it's going more in the sort of the structural aspects of this. Okay, I just wanted to give credit to, yeah, go ahead.
There is a question.
Yes.
Two questions. Okay, first question. When you have a nanospring to measure protein forces, does the spring tension affect the tension the protein has to overcome?
Well, the way I see it is that this nanospring is actually, I didn't put the reference here. It's a Japanese group who invented this, and I should have put it here. I apologize for that. But what you do is you attach one end of this nanospring to the glass surface and the other end to molecules that then interact with the microtubule. And as soon as the microtubule starts to shrink, it pulls the nanospring, and then every component in that system feels the same force. So there's a force that tries to pull the NDC-80 away from the microtubule, which is the same force that is resisting the depolymerization of the microtubule. So it's just action-reaction. So it's all under tension. And it's because the microtubule shrinks and the NDC-80 molecules do not let go, at least if you have sufficient number of them, that you can create this tension on the whole system. And then what we look at, what you see is that if you have if you have just one or two NDC-80s, then at a very modest force, the connection between the protein and the microtubule is lost and the spring relaxes. But if you have many of them, then you will actually slow down the shrinkage of the microtubule up to the point that at some point it switches back to growing. So we call that a rescue. And we can really test how the number of NDC-80 molecules affects the probability to have a rescue and at what force this typically happens. So that's the kind of experiments we are doing. I hope that answers the question.
Yes, I think you did. The second question is, for measuring the force magnitude, do you use any model? I mean, how do you make sure that X amount of stretching gives N Newton of force?
Yeah, a very good point. So we calibrate the nanospring with an optical tweezer. So you have to be sure that that the nanospring, the way it's designed. And again, I really apologize in this overview slide that I didn't put the original reference, but you can convince yourself that the nanospring is very reproducible in terms of its structure. And then instead of putting it on one end, the NDC-80 molecules in the microtubule, you attach the spring to a bead, and then with an optical tweezer you pull, and you can then calibrate what length corresponds to what force. And it's relatively mild forces that these nanosprings, so maybe up to 10 piconewton, this is a good system. If you want to go to very high forces, you just completely stretch the nanospring, and it's not a good system. So it's a good for the sort of the force range from a few piconewton to maybe 10 piconewton. But it has to absolutely be calibrated, yes. And it's not very precise. An optical tweezer is more precise, but for the purpose of where we use it, that's okay. Yeah.
Thank you so much. And I think I dug up the original reference you mentioned, and I just put it in chat for anyone who wants to.
Yeah, that's probably, I'm just gonna see. Yes, Iwaki, that's it. Yeah. So that's where this is the original reference. And we also published actually a paper about this. Now I have to get back to my slides. Oh, yeah. And we have a paper published where we explain how we calibrate. It's not yet doing a lot of biology yet, but at least using this as a spring for this kind of experiments has been published. Oh. Okay, and then on to yeah. So all this kind of experience that we have with minimal systems for eukaryotic cytoskeleton and spindles, we now try to transfer to designing a building and I say optocontrolled. I will explain why. But first of all, a minimal DNA segregation system for a synthetic cell. The reason, and I'll come back to it, there's optocontrolled written here that we would ideally also like to have a way of imposing some kind of a cell cycle dynamics by making the connection between DNA and the cytoskeletal system light sensitive, but I will get to that. And I'm just showing this picture is actually a sort of a schematic that we sometimes discussed within our basic consortium in the Netherlands of, okay, if we want to build from the bottom up different modules that will lead to some kind of a minimal system that is able to not only grow and replicate DNA, but also divide, we need to think about many modules, and you can think about, you need a container like a lipid vesicle that can grow, that contains DNA that it has to process somehow, replicate and translate to protein. And then DNA needs to be segregated once it's replicated. And then the biggest challenge actually is to then make sure that these vesicles, once they've grown and segregated their DNA in some form, divide. And you can do that with spontaneous mechanisms by playing with the curvature and lipid composition, or you can make it very complicated involving proteins like many cells do. And within our consortium, different people focus on different aspects of this schematic. And of course, you can have very many different versions of this, but our contribution is about finding a way to segregate DNA actively, because you could also imagine that simple entropic forces or putting your code on many different plasmids would not even require an active segregation system for DNA, but that's what we are working on. So we are trying multiple things, and we are focusing on finding a way to actively segregate the DNA.
Question
so,
before
yes,
before you move on.
Yeah.
When you plan bottom-up cell assembly, do you only take into consideration systems based on natural components like lipids and proteins, or do you use tools not found in natural biology, like hydrogels or polymers?
Yeah, also very nice question. So I think you could do anything you want as long as it works. In our consortium, we've made the choice to focus on existing biomolecules, so proteins, lipids, DNA. Although I don't think everybody feels that it's forbidden to maybe try something hybrid and use DNA origami if it works. So our starting point is natural systems, but not necessarily one natural cell. So you get inspired by solutions that different biological systems have for different parts of this module. So Bert Polman is working on ATP synthesis, so is using natural proteins, but not in a way that a natural bacteria it would do. So, but yes, using biochemistry as biology gives it to us. Yeah. And for if that's okay, so the active segregation, we also get inspired by how bacteria do this, even though, again, we don't have to exactly copy. And even in the natural world, there's different ways in which DNA can be segregated. So if you have multiplasmid systems, for example, that simply dividing it more or less in two could be enough. There's a plasmid like the PARM system that segregates actively plasmids, but then there's also the idea that the chromosome itself, that is very big, that upon replication simply by entropic forces in a confined space is also a viable mechanism to segregate DNA. We are, like I said, focused on the active system, and then we've focused our efforts mostly on the PAR-MRC system, which is an anti-parallel bundle of PAR-M filaments that pushes DNA apart with the help of a connector molecule called PAR-R. But there are also tubulin-like systems like the TUP-CRC, where you also have a connector molecule connecting DNA to tubulin-like structures, thereby pulling on the DNA. And we've actually tried multiple, but sort of converged on PAR-MRC, at least for now, as being the most reliable candidate or the most interesting candidate. Meanwhile, my family here is coming home, so you could hear some noise every now and then. It's sort of dinner time here. And like I said, so what we're aiming for is be inspired by these bundled or linear filament systems that hook up to DNA somehow. And there's some extra requirements. We want this system to be, we need to be able to express it in pure, because as a consortium we work with the pure system. That, for example, with tubulin from eukaryotic systems is not possible. And then we are also thinking of having a simple system that allows us to connect filaments to DNA, but then engineer the connector molecule. And at that point, of course, it's not natural anymore. to become light sensitive so that we can also dictate when does the connection between DNA and filaments happen, so that we have a way later on to, for example, coordinate with DNA replication or cell division in time. Eventually, we are looking for a biochemical clock or something to regulate this connection, but initially we will, or we want to, it's not working yet, would like to have an external way of controlling when segregation takes place by using light. So our initial was to,
so,
yeah, go ahead.
Question before you move on. How do you attach the protein to DNA?
Yeah, so we use the natural, I will come back to that. We use a natural system that, for example, PAR-MRC. PAR-M is a cytoskeletal filament. It's a bit like actin, a double helix. PAR-C is the code for the DNA that is recognized by PAR-R. And PAR-R is a protein that can bind on one side to the filament and on the other side to the PAR-C, which is a specific code in the DNA. So it's a one protein connector molecule that allows, and that's what bacteria use to connect DNA to the filaments. Unlike kinetochores in eukaryotes, where you need tons of different proteins. Let me just close the door, because otherwise the dog just opened it. Okay. Yeah, and I will get to that. So from now on, it's slides of different things that we tried and ran into different issues, which in the end made us choose PAR-M, even though it's also still work in progress. So we initially thought, okay, we know everything about eukaryotic microtubules. Bacteria also have microtubule-like structures. They are very similar in behavior, but just fewer protofilaments. And we actually did a biophysical study of these bacterial microtubules. They have dynamic instability. We even measured their forces. But we also thought it could be a good candidate to use for the minimal segregation system, even though that's not what it's used for in bacteria. And there's no natural molecule that allows us to connect these filaments to DNA, which is why in the end we also didn't use it. But for a while we thought, well, we will solve this somehow and find a way. But one thing that did work very well is that these bacterial microtubules can be expressed in pure, even inside vesicles. So you encapsulate the DNA, a plasmid that codes for bacterial microtubules. It's actually two proteins, bacterial tubulin A and B, just like eukaryotic ones. Filaments, so the proteins get expressed in pure. They even form filaments and they deform the vesicles into elongated shapes. So that's all very nice, because if you could also connect these filaments to DNA, then you could have a module that not only segregates DNA, but also helps to shape the vesicles in elongated shapes, which may help the division process. But what we ran into, as I said, we could not find a way to connect it to DNA. There is a protein called bacterial B-type-C that binds to the lattice of microtubules, but we could never find a way to reliably bind it to DNA. We also, and here I'm going a bit quick, because, and I'm not showing you everything. If you want to know more details of what we tried and didn't work, we will always be happy to share. I'm just giving you the quick version here. And we also tried the TUP-CRC. That's actually a system that is used, as I explained earlier, by bacterial systems to bind to DNA and pull on it. And here we could indeed show, this is filaments of TUP-C that we purified that form under the microscope. And we could even show that TUP-C, which again is a piece of DNA, in the presence of the connector molecule TUP-R, binds to these filaments and it even gets pulled on. But we were unable to express this in pure, or we could express it, but it was never functional. So again, we for now at least abandoned this system. So one was expressible in pure, the other was able to bind to DNA. So we also tried sort of the hybrid system, where we thought, okay, if we have, for example, we break up the connector molecule, the TUP-R, into two pieces, and that we connect with an SSPB islet. That's for now always the strategy that we have. So we take the peptide that normally binds the filament to the DNA, break it in two, and then get a light-sensitive dimer in between. Then we don't have to use the DNA, and we could use the top R part from the I should say it correctly. Yeah? So we could take the part that binds to DNA from this system, from TAP-CRC, and take the part that binds to the microtubules from another system that is expressible and make a hybrid, because since we're breaking up the peptide anyway, we could take half of the connecting molecule from one system and half of the connecting molecule from another system. But for various reasons that I will not all go into, and I also don't all of them remember right now, we were never able to get DNA bound to the filaments in a way that we were satisfied with. So all this to say that that we then now focus our efforts on PAR-M, also because it was already known that this can be expressed in bacteria. Christophe Danelon's group showed that. I don't know if it was published, but at least in the lab, we knew this already. And somebody was asking, so here's a bit more structural information of how this connection works in the PAR-M system. So in cells, but also you can do that in vitro. And this has been shown by several groups already, that you can grow PAR-M filaments in vitro. They will make anti-parallel bundles. And PAR-R, which is the connector molecule, will bind to the outside of these anti-parallel bundles. And then several of these PAR-R molecules have to come together to bind and curve a piece of DNA. And this is the PAR-C code that I explained. So it's not a single protein to a specific code on the DNA. It's actually several. I don't think it's known exactly how many you need minimum, but the DNA gets bent a little bit, and then several PAR-R molecules together form the connection between the filament and the DNA. And this in vitro works quite nicely. This is not something we showed. It was already known before that you see a movie here that really DNA can be traveling at the ends of these filaments. Since they form bundles, the PAR-M filaments, it appears as if pieces of DNA are also traveling along the bundles, but we think, and others also think, that there's just ends, several ends of filaments in these bundles where the DNA binds and can be moved. And if you then increase, in this case, I think the PAR-M concentration, you can really get very large bundles with DNA attached to it. So the wild type version here works. And now the question is, can we get this pure expressed? Can it be active when it's pure expressed? Can we encapsulate it in vesicles? And what we spent most of the time on so far, can we find a way to break up the PAR-R molecule and make its activity basically light sensitive? Okay. Let me check. Yeah.
There's a new question.
Yeah.
Why do you have to work in pure if whole cell lysate yields are higher?
Yeah, so this is a consortium discussion. So yeah, so I'm not a real expert there, and I rely on others in the consortium who have this experience. So we have at least agreed to for now work as much as possible, everybody in the pure system, the same system. But again, we've not blocked the way to maybe one day switch to cell lysates. Yeah, we could have a long discussion of the pro and cons. I guess the lysates, we would need to have it available in large quantities so that everybody in the consortium can use the same. Willem Huck's group is still working with the lysates, so maybe we'll switch at some point. For now, this is the agreement we came to, even though many of you in the audience probably know much better what are the also the disadvantages of using pure. Yeah. Okay, so ideally one day the idea would be, and this is just a total what I've already said this, but now just in a fancy picture, the idea would be that you have PAR-M filaments, the DNA plasmid with the PAR-C codes embedded in it. Then PAR-R molecules where we take the N-terminus and connect it to islet, for example, and then we take the C-terminus, which interacts with the filament and connect it to an SSPB. And then without any light, they should not interact, and then you get a conformational change of the islet, which is a love domain when you do shine light, and that would then allow for the interaction to happen. And ideally, if you turn it off, the conformational change would reverse and you let go of the DNA. Like I said, this is just work in process, something that we're trying. We've tried actually many ways. So of course you take the PAR protein and you have to choose where to cut it and then where to insert these dimerization domains. We've tried many examples. I'm just showing you one where it's called a new optogenetic construct, because for us it was new, we've tried many before. And this one also makes artificial dimers of the PAR protein such that we have multiple molecules together interacting with the DNA. And the short story so far is that we can make this work, but not light sensitive. So we can make these constructs, purify them, even put fluorescence proteins in them. And they will come together in such a way that we can show that the DNA actually gets recruited to the filaments, but it doesn't let go when we turn the light off. That's where we are for now. And again, if you want to know more details, we would be happy to share with the people who actually do this in the lab. I think I have another movie just showing that the DNA transport works, and this is not yet in a vesicle, but it is two peptides that we've designed that have the islet and SSPB construct in them that for the moment bind very tightly, even if independent of whether we turn the light on and off. And what we're trying is, and together with a colleague, new assistant professor in Delft, is to use tools like AlphaFold to maybe predict where we can insert the love domain or make the construct in a slightly different way to make, probably the affinity is just not, between light and on and off, there's not enough distinction for us to be useful. It could also be that, that this is just the wrong strategy, and we're always looking for advice and people who have ideas about this. But this is more or less what we are.
Speaking of that, a question is, have you tried chemically induced proximity instead?
No, but we've talked about it, and maybe it's one of the things we've, so not enough yet, but it's one of the ideas, I guess. And I don't know who's asking the question, if he or she has a specific system in mind that I could put in the chat, then it might help us. No. I mean, there's also systems that bind to DNA that dimerize upon the presence of light. So that is an like Serafina Wegner in Germany has experience with that. So we have a few other constructs in the freezer that we haven't tried yet based on some other ideas. Like I said, this is really, we're in the middle of it. And what you should really do is invite Beatrice, who I will show her picture in a second, who is doing these experiments. So I'm sure she would be happy to also contribute, if that's of interest, some of the details of all the different things that we've tried and are trying.
So the person that asked that question did answer your request. He said we used Catchfire system.
Okay. Well, thanks. That's this is just a picture that I'm not going to dwell on. This is what I said. So using structural computational biology with one of our assistant professors, we're trying, at least in the computer, to insert the domains in various places to see what might be the best bet. And then we also think, so there's different chemical approaches. And then another way to, at least for now, control par-M polymerization is simply to use caged ATP. And that's fine to turn things on, but then of course you cannot use this, I assume, unless you become fancy about reproducing caged ATP. You cannot cycle this interaction, but at least this works. We can, by shining light in the presence of caged ATP, we can really trigger the polymerization of the filaments and the connection to DNA. So that's something, a sort of intermediate that we may try. Okay, I have a few more slides and then I will stop and see if there are more questions. One is just to, so this is all about trying to get opto control, but whether it will work this way or some other ways, we also need to make sure that, that it actually works in pure. So this is just some pictures either on SLBs or no SLBs to show that, that if you have, that if you take purified par-M, which everything I've shown you so far is done with purified proteins, if you put that in a pure solution, it will polymerize. So the conditions in pure are fine for polymerization of par-M. I'm not showing it, but when we express pure, par-M, sorry, and par-R as well, in pure, we can see on a gel and that it gets expressed and also at fairly okay concentrations, but it's not really polymerizing, at least not when we express for a very long time. Meanwhile, we found that if you put purified par-M in a pure solution that has been sitting around for many hours, the time that you would usually use to express it, it also doesn't polymerize anymore. So it's not, and so we now have to check, make time series of whether when we express in pure MR, is there a time window where it will polymerize? Because what we've seen so far, that if you wait too long, something happens to, I don't know what, and other people maybe do know, where that doesn't support polymerization anymore. So I'm now really showing you what sort of happening on a day by day. And okay, this was the purified par-M, but like I said, we tried muscle cellulose at different temperatures. But I believe all of this was after a sufficient or quite a long time of expression in the pure system, where then polymerization is not supported anymore. But as is not shown in this slide, but I said earlier, actually also not for purified par-M anymore. So that's where we are. Okay, I didn't want to take a huge amount of time, so I'll just finish by also introducing the people who are currently working on this project. So Yash and Beatrice here. Yash was the first to start. He did, together with a former student, Reza, many of these experiments with bacterial microtubules and TUP MRC. Since we've switched to par-MRC mostly completely, it's Beatrice, who is now halfway her PhD, who is also doing this collaboration with the structural biologists to see if we can find other ways of inserting the love domains. And very recently, Yu Cheng, coming from Shenzhen, joined us about a month ago. And he will also be within the EVOLVE project, not only helping to get this DNA segregation system, whether it will be optocontrolled or not, but get it in vesicles and working in the pure system. And then one of his tasks will be to connect to DNA replication. So other people in the consortium are in the same system building DNA replication modules. So we would like to connect to that. And then specifically with Gijsje Koendrink, who I believe is speaking next week, if I'm informed correctly. She has a role in this consortium more on cell division and shape control. So there's now 2 students in her group, Joris and Lisa, who started very recently. And so they will work together as a team to not only get individual modules to work, but also in the same system, get at least multiple modules connected to each other, which is why this time control is something that is of interest to us. And then the final person I want to introduce here is Olga. And she is, yeah, we also have an effort, as you know, in ethics and philosophy in this project. And she did a very nice study that was also recently published, where she asked in many countries in Europe, had a survey which was about what the general public, and I'm probably not saying it completely correctly, but thinks about different kinds of applications of synthetic cells. So once we have them and we use them in a medical context or for materials, or what people think about it, and she had to translate all these questions in I don't know how many languages. And then when all the answers came back, she had to translate back from all these different languages. And published about the results of it also not very long ago. It's a collaboration with Huub Zwart in the Rotterdam, Erasmus University in Rotterdam. So these are the people currently working on the Evolve project together with Gijsje, but of course I'm not listing everybody. Other groups are working on DNA replication and cell division, and you can just go to the Evolve website to find out. This is a bit of advertising for the basic program, where we've built different kinds of modules so far, and now we've switched to this Evolve program that you can find online all about. And there's actually more PIs now involved than in the previous program. If you're interested, you can look it up. And then maybe the final slide is, since I've introduced everybody in my group, this is of course the most important people. So this is technical support that is often shared with other groups. So Eli, Ashmiani, Singul, and Roland, they support us in protein purification, lab management, microscopy, often giving the same kind of support to at least one other group in the department. So with that, I think I've thanked everybody in the group indirectly, and just see if you have more questions and put them back up here. And thanks for having me again. And
Thank you. Thank you so much. There are a few more questions in chat. So the next question is...
Stop the share, is that okay?
Yeah. Oh, then you can see chat yourself.
Oh, yeah.
So I think now you're at Adam's question.
Oh, the dog should join the call. Yeah, well, that was not a question, I guess. Let's see. The catch fire system, and then does vesicle composition affect your consideration to? Yeah, no, I mean, this is yeah, it makes a lot of difference. So yeah, so I'm what we've tried the most, where we saw that this makes a big difference, but I think everybody who works with vesicles knows that it makes a difference. And then depending which protein it's not like you take one composition and then it works for all the proteins. It's really tricky. Here I also depend a lot on my colleagues who are more experienced with this, that making vesicles you can make with uncharged with DOPC, but as soon as you have proteins in there, it matters sometimes if you have charged lipids or not. So whether we haven't settled on a agreed upon composition of the lipids for the consortium for now. I mean, people in Groningen who are doing ATP synthesis, they need also transmembrane proteins. So it's a whole art on its own. So it makes a lot of difference. With tubulin, I don't know if anybody, we have a lot of trouble. I mean, there's all the different methods of making vesicles, like C-dyes, E-dyes, emulsion-based transform methods. And whenever we do, we find the composition that works okay-ish for bacterial microtubules or for actin, then we try it with tubulin, and it's almost as if tubulin itself acts as a lipid and goes to the interface, goes into the oil. So it's something we simply haven't solved. Yeah. And I know it's a very vague and not so helpful answer, but it's, I think, the reality. Yeah, so the DNA replication, so 529 is what Christophe Danelon did. And we, in the consortium now, we also have Gijs Wuiten, who's working on the T7 system. So again, it's not something we are doing. Yeah, so if I remember it correctly, the T7 system replicates circular DNA. It is 4 components I think you need to, it's the helicase, the polymerase, and then you need additional proteins to resolve the plasmids like a topoisomerase. So to make that work, you eventually you need not only a replication system, but if it's a replication system that works on a circular DNA, you need a few extra proteins to also be able to separate the DNA. So this is one of the other modules that we are not working on, but collaborating eventually to combine. What kind of technique have you used for the vesicles? Yeah, so what I was showing, so here were mostly droplets, and then we use different techniques. What we've tried for the vesicles, especially with tubulin, but that we are now also, I didn't show you, but we've tried to encapsulate PAR-M already in vesicles, and that is okay. So it's most of the time an emulsion transfer based method. So this can be very simple, where you take oil and aqueous solution with your proteins in it, and you shake it, or you drag it over a rack, and then let this emulsion sink through a second interface, or centrifuge through a second interface, or you can use microfluidic techniques to achieve the same goal. All that works. Different people have different how do you say, preferences. If you do it in a microfluidic device, you have more control over the size, and it's more homogeneous size distribution. If you do it with pipetting or some other way of making emulsions, you have less control over the size, which sometimes is an advantage if you don't want to fix the size. The biggest trouble starts with when you put in proteins, and which proteins. With relatively forgiving buffers, all of these methods work fine, but as soon as you put in proteins, it's more difficult. But tubulin seems to be particularly complicated. With most bacterial microtubules, for example, we have what you would think is quite similar, but we have no problem using C dyes and E dyes emulsion transfer methods to get the protein in. And the other way is to do swelling, but mostly we do an emulsion transfer.
Thank you so much. I think that's all the questions that were in chat. I was really interested to hear about the T7 system, because we've been using 529 too, and we have problems with those, not problems, but just the reality of those giant concatenators being the product. So I'm really looking forward to learning more about that.
Yeah, and this is, we just had we have now, of course, different, within the Evolve program, we also have now a new organization. It doesn't really matter, of meeting in different compositions of groups. And just last week we had this discussion about the T7 system, but it's also a new student who just started with this. So we'll see where that goes and how we can combine. Yeah.
Thank you so much. Any last questions? And if not, I want to say thank you so much, Marileen, again, for taking the time to give the talk.
Yeah, thanks. And it's nice. It feels like it's still an informal discussion, even though I don't know in how many different places of the world we are, but thanks for being here. Yeah. And I see Hagen behind his desk. Hi, Hagen.
Thanks, everyone, and have a great rest of your Monday.
Okay.
Bye.
Bye.